Welcome to the RIEL Seminar Series. We are well into May now, starting a beautiful dry season. I will start off with some quick housekeeping, just for anyone that's not familiar. We do have the toilets just out the door and to the right there, and in the case of an emergency, we will evacuate out onto the grassy breezeway. For people joining us online, please keep your mics on mute and your cameras off until we get to question time. And I just wanted to remind everybody that today's seminar is being recorded. So please let us know if you're not comfortable with being in the recording. Too bad. Now, I am going to hand over to Professor Alan Anderson to introduce today's speaker. Thanks, Alan.
Just like to start by acknowledging we're on Larrakia country, beautiful Larrakia country. I just want to provide a little bit of a context to Ally's PhD, because it's very much a story of serendipity. So I've been working on answers indicators of restoration success at Ranger uranium mines since the early 90s. Much of it has been in collaboration with the Office of the Supervising Scientist, which is a federal agency that was established to look after the environmental performance of uranium mining in the Alligator Rivers region. And a successful restoration of ant communities is one of the success criteria for successful ranger restoration. And back in October 2021, my OSS colleagues asked me to come over for a chat of their thoughts on what's the next steps for advancing ants as indicators of restoration success in the Alligator Rivers region. So I went across and they said, Alan, so we've established this new genomics capability and we want to explore the use of eDNA in particular for ongoing monitoring of ants at Ranger. And we would like to fund a student to do this, and we're hoping that you'd supervise them and be able to find a student. And I said, well, yeah, no, very interesting, happy to supervise it, but I've got no students in mind. I don't know anyone who does genomics, but look, I'll just let you know if I do find one. And the next day, I happened to be in the tea room and Ally was there making a cup of coffee or tea. And so I'd seen Ally just sort of walking along, never knew who she was. And so we just started talking, saying, oh, Ally, yeah, what do you do? Oh, no, I've just finished a master's on environmental DNA and I've been doing some casual work on it. I go, what? Would you like to do a PhD on? And it was actually ants and termites. Yeah, and so that's how it happened. And Ally's done, as you'll hear, an absolutely brilliant job with it, and just some fabulous results. So over to you now.
Thank you, Alan, and thanks everyone for coming today. So my PhD, as Alan kindly introduced, is looking at genomics approaches for monitoring terrestrial invertebrates, particularly ants and termites, as bioindicators of ecosystem change. And I'd firstly just like to acknowledge that some of the fieldwork and research was done on Larrakia, Koongarra, Kani and Madjawa people's lands, and I pay my respects to the elders past, present and emerging. So why are we doing genomics approaches for monitoring? Well, firstly, there are a bunch of challenges with biomonitoring in the first place. Firstly, for invertebrates, you normally have to do a morphological identification, which takes a lot of taxonomic expertise and time. And there's also a significant trapping effort that you have to do when you collect these specimens. And there's me in the lab doing some sorting, and it's quite time consuming pulling out all the little ants out of your pitfall traps. So I'll go through a few definitions that I'll talk about in the talk today. The first one is barcode. And when we talk about genomics approaches, barcode refer to a vouchered specimen. And this vouchered specimen is normally housed in a museum or a reference repository where you do your DNA extraction on a taxonomically verified voucher specimen, and you get your barcode at the end. And this barcode is basically, you can think of it like when you go into a shopping centre and you look at all the different milks, and you can kind of scan and see and product information when you do your scanning. So for our DNA sequence, it's basically just a series of A, T, C's and G's combined to form that barcode. So when I talk about metabarcoding, basically we start off with mixed specimens. So we don't really know what we're starting off with, and we do a DNA extraction on that, and we get several barcodes at the end. So these barcodes we can then match to our vouchered specimens using bioinformatics and what we call amplicons. So before we get to amplicons, we have to use this thing called primers. And primers are basically short sequences that you attach to the start and the end of your amplicons that bind to the conserved regions across your barcode. And another question that normally comes up is, why environmental DNA? Well, and what is environmental DNA in the first place? So environmental DNA is basically DNA that all living organisms leave traces of in their environment. So as they're interacting with their environment, like a lovely NT billabong here, you can see that birds flying through will potentially leave DNA in the air. Barramundi swimming in the lagoon may leave their DNA in the water, and other organisms may leave their DNA in the sediment as it deposits over time. And also a lovely picture of some ant mounds and some termite mounds. You can also potentially collect DNA from there as well. So we have biotic sources, which we can come from your plants, gut, invertebrates, and gut contents of these animals. And we also have abiotic sources. So when we do our sampling, we can do our sampling when these animals aren't present and still see what communities have been moving through an environment. So when we talk about a metabarcoding workflow, generally we start off with our sample collection, which is our specimen or bulk sample. And then we also have our eDNA samples. So in this example, we have soil and water. We then move on to our DNA extraction. And the DNA extraction, we can then add tags before we do our PCR, so we can match the exact sample back to your original individual sample. Before we do the sequencing, which is on a fancy machine, and the bioinformatics, which we can then match to our vouchered specimens to look at an analysis of ecological signals. So why ants, termites, and eDNA? Well, as I mentioned before, there are several biomonitoring challenges, and one of them is restoration monitoring. So we want to look at how ecosystems are recovering over time. And ants and termites have been identified as key bioindicator taxa for Ranger, but also the reason why they are selected is because they're abundant, responsive, and for ants, widely used as a bioindicator already. But for termites, they're underused, but they play a really key role in environmental conditions such as decomposition and other soil processes like nutrient cycling as well. So it's really useful to see how those species are coming back post-disturbance. So as I mentioned before, we have several biomonitoring challenges with identifying to species. For ants, for example, there are approximately 5,000 plus species across Northern Australia, so that's a lot of species. And for termites, we have about 50 in the Northern Territory. So it's a lot smaller species lists, but still very, very difficult to identify under a microscope. So when we do the DNA and eDNA metabarcoding of our samples, what we're hoping to get is practical, repeatable, and accessible monitoring, particularly important for long-term biomonitoring of any sort of regulatory framework. As Alan mentioned earlier, so this work was sort of done to try and eventually look at Ranger uranium mine, which surrounds Kakadu National Park in the Alligator Rivers region, and ants and termites have been chosen as bioindicators, and part of the closure criteria of the mine is to look at species composition, species richness, and abundance of these taxa. And we want to see if the rehabilitated areas are similar to or on a trajectory towards that of its reference ecosystem, which is Kakadu National Park. So some of the research gaps I identified when first looking into this, because nobody's sort of done it, that's the whole premise of a PhD, you kind of look at what has been done already for terrestrial metabarcoding, and I found that it was a lot less developed than aquatic metabarcoding. And particularly for ants and termites, ants tend to forage a lot on the soil, what we call epigeic ants, but they also exist in other parts of your ecosystem. So you can have arboreal or subterranean, but most of the species composition that you could use for looking at reference sites, it's a lot easier to do with pitfall trapping in soil. So soil was identified as a key substrate for ants. And termites, we had another student, Ellen, who identified that bait substrates worked really well for sampling for termites, and I've kind of gone down that path. But before doing that, I also looked at certain research gaps that were present in the literature. There wasn't any taxon-specific primers that were validated for ants and termites. There were reference sequences that were incomplete for both taxa, and nobody's really looked at eDNA versus morphology concordance for these species, or for these taxa. Also, what I found was that there was extracellular DNA persistence was unknown for these substrates. So when ants are walking along the surface of the soil, we don't really know how they're shedding, but they do shed eDNA, as I'm about to show you. But we don't really know how long it lasts in the soil, and there are certain other abiotic factors, biotic and abiotic factors that influence the persistence in soil. And this limits, all of these limit reliable monitoring in its current form. So my thesis aim was to look at developing robust genomics approaches to monitoring ants and termites as bioindicators of ecosystem change. And I looked at 3 key questions to do that. The first one was to look at to what extent do newly developed taxon-specific primers improve metabarcoding and performance of ants and termites across specimen and eDNA substrates. The second question was looking at can DNA metabarcoding of bulk samples recover similar community composition and site differentiation to morphology in a mine site revegetation context. And the third one was to look at can soil and termite bay eDNA reliably detect variation in ant and termite assemblages in relation to long-term fire regimes. So to do that, I looked at doing several different data chapters, which I'll talk through. So the first of which was validating CO1 metabarcoded primers for ant monitoring, which has recently been published in the journal Environmental DNA. And before starting this, these guidelines came out. So Australian government employees and also other eDNA scientists in Australia sort of got together and came up with the first sort of Australian eDNA test validation guidelines. And within that, they described if you were to do metabarcoding on your samples, these are the guidelines that you should follow. And so within that, I developed a hierarchical validation workflow, which looked at in silico, in vitro, and in situ process of elimination. So starting off with in silico, to develop new metabarcoding primers for ants, you firstly do it on the computer to see how your primers are able to amplify ant DNA, and then you move on to the lab. So the in silico process of elimination went from 20 CO1 primers or 14 primer sets down to the 3 best performing assays. So the in vitro then did a series of experiments in the lab, which then took it down to 2 assays. And the in situ, meaning in the field, I used a case study at the Terrestrial Wildlife Park, which looked at historical community composition, comparing ant species composition that a previous student, Francois Brassard, had done. And I took 5 soil samples from those same plots and compared the two. So Francois did 3 sampling events over several years or several seasons, which resulted in 90 pitfall traps, whereas I only took 5 surface soil samples. So it was validating both the new ant metabarcoding primers and the previously validated primers for terrestrial arthropods, and seeing how they worked across a series of experiments. So the conclusion and the primer recommendations. So for ants, what we found was that the ant-specific metabarcoding primer that I developed in this study was able to both do soil eDNA surveys and work well with specimen metabarcoding as well. Whereas the general arthropod primer that survived the end of that process of elimination workflow wasn't recommended for soil eDNA surveys because the ant metabarcoding primer worked better, but it was still recommended for specimen metabarcoding. And the next data chapter also looked at termites, developing new metabarcoding primers for termites, but instead of the CO1 gene, I looked at the CO2 gene. And previous literature has identified the CO2 gene as being more taxonomically discriminative for termite species. So there were no previous metabarcoding primers validated for the termite CO2 gene. So I had to design all of these from scratch. And instead of soil eDNA like I did for the ants, I looked at termite sheeting, which is basically this material that termites build or construct when they're eating a bait or eating them or building their mound. It's a way for them to build structure, for them to walk through the tunnels while they're eating. Whether it's grass, like underneath grass, and to protect themselves from the UV. And so termite sheeting was found to be successful for eDNA metabarcoding using our primers. And I found that one primer set actually worked really, really well for both specimen metabarcoding and eDNA metabarcoding. So this paper hasn't been published yet, but it's due to be submitted very soon. So great, now we have validated primers. How can we use this to answer our ecological questions, particularly around how we can use ants and termites as bioindicators of ecosystem change? So one of my next chapters looked at specimen metabarcoding, and firstly, we started off with using a different mine in the alligator rivers regions, different to Ranger uranium mine. So Nabarlek uranium mine has been revegetated from 1995. So it had been a long time since revegetation of the mine. And we had different disturbance categories across the mine site. We had our reference sites, which are the undisturbed. We had our pits and evaporation ponds, which are our disturbed sites. And there was also one irrigated site. So the irrigated site was irrigated with water from the evaporation ponds into the surrounding savanna. And since it had been a long time, we wanted to see how the ants and termite communities have come back to the reference sites or haven't come back. And the sampling workflow for this was looking at pitfall traps for ants. So I laid out about 260 pitfall traps and about 240 toilet paper baits. And then that took several field seasons, actually. That was quite difficult. It involved a bit of helicopter work because it turns out that all the roads are closed during the wet season, and Cahill's as well is closed. So yeah, quite difficult, quite hot. I didn't mention this in the previous chapter, but termites are most active in the wet season So you have to sort of do the sampling around then when they're more likely to be attracted to the baits. Whereas ants, you can sample in the dry season, and they're not affected by seasonal variation. And in fact, actually, wet season sampling is worse for ants because water can fall into your pitfall traps and everything can escape. So the dry season is actually the best. And then, so we took all of these samples and looked at them under the microscope. So for ants, each pitfall trap could have about sometimes even 10 species per trap and multiple of those species in each trap. So we sort of set a cutoff of about 50, I think, 50 ants of the same species per trap. And we had a lot of ants. So we identified them under the microscope, which took months and months and months. And termites, luckily only, well, not luckily, but out of the 240 baits that I set, only about 52 of them actually had termites on them. So it wasn't very high reward for the effort that you put in. Whereas for ants, 260 traps were laid out. We had 250 pitfall trap samples in the end with ants. So quite a higher success rate compared to termites in terms of the sampling effort and the samples that you get out of it. So for morphology, we had this reference ID's original data set. So after doing the morphology, we said, okay, these are all the species in each of our traps. And now we're going to compare it to the metabarcoding. So for the metabarcoding, I decided to do a non-destructive extraction technique, which basically keeps the specimens from being destroyed. So you don't actually have to crush everything up in a mortar and pestle and get out the DNA. You can actually extract the DNA out of these organisms and keep the exoskeleton of the insects intact. And one of the ways that I looked at doing this was also doing some size sorting while doing the DNA extraction. So the size sorting was to help with this biomass issue where larger ants are an order of magnitude larger than the smallest ant. And this biomass can cause a swamping effect where the smallest of the ant species isn't picked up by metabarcoding because of the amount of DNA that is extracted from these larger specimens. So for ants, there was several different size sortings, and for termites, we just had soldiers and workers. One thing I will say about termites, and for ants, actually, just going back. So ants, you can identify from just the workers. You don't need, there's just workers that you get in your pitfall traps usually. You get the flying alates only very rarely, but you identify normally from the workers. Whereas termites, you actually do your identifications on the soldiers, and you can't really identify from the workers. You can if you extract the guts out of the termites, but we don't really want to do any of that. And yeah, so you really need the soldiers. And to find the soldiers on the termite baits, it's usually 1 soldier to 100 workers. So it's quite difficult and time consuming. You can spend about 20 minutes on a bait trying to find the soldiers. So following the size sorting, I also pulled the DNA equi using an equimolar ratio to help with this biomass swamping issue before doing my PCR sequencing and coming up with my metabarcoding data sets at the end. So this comparison framework between the morphology and the metabarcoding allowed me to see how well the metabarcoding actually picked up the species that we were interested in, and seeing how well we can potentially substitute metabarcoding for your instead of using morphology. So the research questions for this chapter were, to what extent can DNA metabarcoding identify potential errors in morphology-based identifications and thus improve reference data sets? And the second research question was, how similar is the composition of ant and termite assemblages as detected by metabarcoding to that shown by morphological identifications at the trap or bait level? And then I wanted to look at to what extent can DNA metabarcoding recover patterns of ant and termite community composition at the site level of a revegetated post-mining landscape across disturbance categories. And then also finally look at how well does primer choice influence detection and taxonomic resolution in bulk ant specimen metabarcoding. So from chapter 2, I'm not sure if you remember, but there was 2 primers that were recommended for specimen metabarcoding. So I really was doing everything twice because we had 2 primers that worked well for the ants and we didn't know which one worked the best. So I really wanted to try and figure out in this chapter which one worked the best. So for the first question, can DNA metabarcoding identify errors and improvements in morphology reference data sets? For ants, what we found was that DNA metabarcoding was able to improve the original morphology data set by identifying that 10.1% of original records were corrected. They were either data entry errors, which happens when you've got so many ants, and also they're quite cryptic. So a lot of Alan and Francois and Benjamin Hoffman's work has been looking at the megadiversity across Northern Australia and based off of genetic location and morphologic data. And the difference between one species and another is lost to me, but they are different species, and it could be that the eye of the monomorium is slightly oval or it's slightly more circular, and well, there you go, 2 species. They look exactly the same. They're 2 millimeters in size and absolutely tiny. So it's not surprising that metabarcoding can actually improve the data set when there is room for human error when you're doing the morphological identifications, particularly in such small organisms that are so abundant and diverse. And for termites, what we found was that the morphology-based identification, when there wasn't any suitable soldier specimens, that metabarcoding was able to recover taxonomic IDs for those specimens at the species or genus level. So that is an improvement because if you were to get this termite morphology data set and you have 10 traps without soldiers, well, then you won't get any species composition data for those traps or those baits because you can't really identify from workers. And looking at how similar the community composition was detected by metabarcoding, sorry if this is a bit hard to see, I'll just change to a laser pointer. So we had our 2 primers that were used for ants. So we had the FWH, which is the broader arthropod primer, and then we had our ant primer or our ant specific primer down the bottom. And for the FWH primer, I'll explain this a bit more. So a Procrustes test looks at a non-metric dimensional scaling, or NMDS, which you might be familiar with community composition. And an NMDS looks at how similar communities are to each other, and this is based at the site level. So at particular points, you'll see a circle, which, for example, is here, and then an arrow. So for the FWH primer, the circle is the morphology and the arrow is the metabarcoding, and the Procrustes lays the NMDS on top of each other and rotates so that you can see how the 2, you can compare the 2 methods with each other. So for the FWH primer or the broader arthropod primer, the Procrustes R was 0.84, which is pretty good. But for the ant primer, it was 0.985, which is even better. So the Procrustes correlation was better with ant primers, and this is at the species level. I might have skipped over this, but the species, so ants are all one family, so you can't really just get to taxonomic assignment at just the family level because that tells you nothing. there are several genera, but even within the genera, there are even more species. So getting to the species level in metabarcoding is really, really difficult, and it depends highly on the reference databases. So Alan's been collecting Tier1 barcodes across Northern Australian ants for a very long time. And in particular for this site, when we were building our reference collection, we made sure to get species level barcodes, and these ants with these barcodes are housed in the magnet collection. And this is why we were able to get such great results in the end, was because we had the barcodes for our species. And this was at the species level, but even at the species complex or the genus level, the ant-specific primer also worked the best, which is great news. Okay, and then we wanted to know, can DNA metabarcoding recover patterns of disturbance that we found existed with the morphology data, but in particular, we wanted to know if metabarcoding also reproduced these patterns. So I might have mentioned at the start, we had our undisturbed sites, which are in green here, and our disturbed sites on the mine, which are in red. And at each point, we have three different shapes, and that corresponds to our methods that we used. So for ants, we had the 2 primers plus morphology, which is why you see 3 different shapes in here on our NMDS. And then for termites, we just have the 2 because we only had 1 primer that we used for termites. And the clustering for the ants for the disturbance categories, the green undisturbed sites cluster separately from the red disturbed sites. So even after, how many years is that? Nearly 30 of revegetation over time, there is still clear community composition differences between the undisturbed and the disturbed, but there is some sites that are closer to the undisturbed sites than others. So that's good news, that the methods for the 3 different primers cluster quite closely together. So there's a line between each of the 3 shapes, which is quite small. If I go over to the termites, for example, and we look at the morphology for this site, FIA2, and the metabarcoding result, the line is quite large, meaning that the community composition between the 2 methods is different. But for ants, what we're finding is that the lines are a lot more clustered, meaning that both of our primers are giving us really good results compared to our morphology. And also for termites, there is no clear clustering of disturbance, and that could just be due to the low sampling numbers that we got from our sampling effort. And the next thing I wanted to look at was, how does primer choice influence taxonomic detection and resolution? So for metabarcoding, what we hopefully want to see is a full match. So we have our reference ID saying it's Monomorium species lay 1, and we have our DNA metabarcoding result saying it's Monomorium species lay 1. That's a perfect match, or a full match. And what I wanted to do was look at developing a scoring system that can see how well our taxonomic resolution is actually getting to the correct ant species. And what we also have sometimes with metabarcoding is an ambiguous species, which is where, say, Nylanderia species 3 obscura group is in our trap, but then the DNA metabarcoding is saying, actually, it's species A. And we wanted to see if that was a correction in our data, or actually the metabarcoding was wrong. So we gave it a different score. And then also for genus, so for example, Iridimemic species 3 for our reference IDs, but the DNA was just giving us Iridimemics, which is still okay. It's still telling us something, but we want it really to try and get to species 3 if it can, because that gives us the best resolution for our data. And if there was any extra hits in our DNA. For example, reference ID said there was nothing, but then for some reason metabarcoding said actually there's species, Nylanderia species A. And also if it was missing, so we gave it a score of -1. So reference ID says there was Tetramorum simillimum, but DNA metabarcoding said there was nothing. And the results are interesting. So the ant primer is on the left, and you can see that its mean is higher in terms of the primer performance score than the FWH or broader arthropod primer. So that's good news. So not only did they recover similar community composition to that of morphological identifications, but also they the ant primer worked better, which I really wanted to know because it's so annoying having to do 2 metabarcoding primers. You're just basically doubling all of your samples at the end, which is quite expensive and a bit annoying. So if you also notice down the bottom, what we found is for the -1 scores, the ant primer had less -1 than the FWH primer. And I will say that the FWH primer is a little bit more broadly used in the literature at the moment because the ant primers didn't exist. So hopefully people take up the ant primers. We found that it's the preferred choice for ant primers are the preferred choice for targeted bulk DNA ant metabarcoding in this system. So it could depend on the system that you're looking at, but for this particular system, using pitfall traps and savanna ants, that's what we're recommending. So the conclusions for chapter 4 was that bulk specimen metabarcoding can detect ecological patterns associated with land use, disturbance, and rehabilitation. It's also a very useful audit tool for morphology-based data sets. So not only does it help correct human errors, but it also helps to differentiate cryptic taxa as well, which can be really difficult even for the experts that when you're looking at so many all the time. And these results support DNA metabarcoding is a robust tool that is better suited to standardized and repeatable long-term biomonitoring when paired with other voucher-based taxonomy and well-curated reference databases. And the last data chapter I had was looking at how well did eDNA metabarcoding of ants and termites recover ecologically meaningful patterns based from specimen-based morphological identification. So we used the Terrestrial Wildlife Park, which is located in Berry Springs here in the NT, and it's a long-term fire experiment, which I think has been running for 22 years this year. And it has a randomized fire treatment design looking at across three replicated treatments. What we found was that for ants and termites, based on historical surveys of research done by Francois Broussard and Ellen Roffelmeyer, who looked at ants and termites at the park across fire treatments, the unburnt, which is fire exclusion for 22 years, has a species composition very different to the burnt early every year for both ants and termites. And what we wanted to look at, well, can eDNA of soil for ants and termite sheeting for termites be able to see those ecologically different patterns just from the eDNA. So we had a sampling plot, 15 pitfall traps or 15 termite toilet paper rolls buried in the park. And for the ants, what we had was using a soil composite sample. So at each point, I had a 20 by 20 centimeter quadrat and took some soil from each to form one composite sample. And for termites, we had buried toilet paper. When there was termite sheeting present, you scrape off the sheeting And we also had field negatives, cold storage until extraction, and pretty strict contamination control, because you can, eDNA is everywhere, as my first slide, one of my first slides showed. So you can really track eDNA around, which can influence some of your results. So for ants, what I found was that species-level ordinations derived from pitfall trapping and soil eDNA showed very strong concordance, which is pretty awesome. At the species level, pitfall-trapped ants assemblages showed a strong treatment effect, which is also great. So not only did we want to know was the species community composition similar, but we also wanted to see if eDNA was able to recover the fire treatment effect that we found with morphological identifications. So that's great news for the ants. Termites was a little bit different. So we had species-level ordinations from specimen sampling and bait eDNA also showed a very high Procrustes concordance, but there was no fire treatment structure like we saw for the ants. So this could be a limitation due to the way that the data we had for the historical termite surveys, we weren't able to separate just bait species composition. It also had mound surveys and active searches that Alan did as well. So that treatment structure wasn't there for fire just yet. So the conclusion for that was that eDNA metabarcoding can recover ecologically meaningful patterns across long-term fire treatments, particularly for ants. But ant eDNA showed really strong species-level concordance and recovered the same fire treatment-driven assemblage gradient, which has clear biomonitoring applications in tropical savannas. And for termites, it did not reliably reproduce species-level treatment differences or compositional patterns detected by specimen-based surveys. So the thesis key takeaways and implications for biomonitoring are that ants are well suited to genomics-based biomonitoring already. So you can definitely use it for either specimen metabarcoding or soil eDNA to look at how species change across your different gradients. But termites remain potentially valuable bioindicators, but genomic implementation needs more work. This could be due to our sampling design or just having a think about how we can get better termite eDNA species composition. And then genomics approaches can broaden access to a terrestrial invertebrate monitoring. So once you have these custom reference libraries or vouchered specimens, more people are able to uptake this work and monitor ecosystems using these taxa. And I'd like to thank Alan, my principal supervisor, Sam and Maxine, co-supervisors, and Andrew and Kate from ERIS, or Office of the Supervising Scientist, for all of your supervision help and contribution to these chapters. And also like to thank all the volunteers, collaborators, and traditional owners, as well as Australian Government, for providing an RTP scholarship, and RIEL for hosting me all these years. And biggest thank you to my friends and family as well for supporting me through this awesome journey.
I hope we all agree. Very impressive body of work, isn't it? And with such important implications for genomics approaches to invertebrate monitoring. Got time for some questions.
I would argue that the termites are potentially not a good eDNA. You know, we often have certain taxa or species that eDNA just doesn't work that well for, and I suspect that based on their ecology and also frequency underground, it's actually going to be very hard to actually use them as a reliable bioindicator. Do you think that way? The ants are really good. The termites.
I have to agree. Yeah, just based on the sampling effort and just. Yeah, even getting termite sheeting, like it can be useful, for example, when termites leave a bait. So sometimes they can come to a bait, eat a bit, leave, and so you don't have any specimens to collect. Like that could be useful, but using it just using eDNA to look at termites as bioindicators, I don't think.
I think it's a good answer. Look, for some who think like, oh, we're going to be able to detect everything. We can't, and there's good reasons why. So termites just might be one of those groups that just doesn't work that well.
And I think also even traditional baiting techniques for termites are not necessarily robust because they just pick up a very small subset of termites anyway. So it's not a question mark.
Are there any literature that suggests that if you did this something again in 20 years' time, that the disturbed and undisturbed plots, I think in chapter 4, would be more similar?
Yes. So that's so what you look at with restoration monitoring is trajectories or restoration trajectories, where you want to see that disturbed sites are becoming more similar to reference sites. So there's been a lot of work in southwest WA and also in the Pilbara, I believe, more southwest WA, showing that sites do come back for invertebrates over time, and they have chronological sequences. So they have scattered years where they have different rehabilitation timelines. So they can see that over time it is actually coming back, whereas we just had the one sampling event and it's really hard to see that.
I'll have a question about that, students. So first of all, a couple of questions. First of all, is that that comparison, is that just species composition?
Just species composition, but I didn't talk about this. So I used frequency of occurrence when I looked at my data as well. So it can be a proxy for abundance because we knew exactly how many ants were in each trap. That potentially could be something that I do talk about in the paper, but didn't talk about here. And you could potentially do occupancy modelling and things from that sort of data. But yeah.
Also, maybe that, as I understand it, so you sampled one natural site.
So there are two reference sites, but yes. Yeah.
What's your feeling just about heterogeneity in the environment? In other words, if you sample 10 natural sites, maybe you would all.
Yeah. No, that is a really good point. Particularly for the irrigated, it really needed its own reference site because the species composition was a bit different compared to the other reference sites. But it is quite patchy. So it's a really small mine, and the sites are close to each other. So it was really hard to select a reference site in the first place, but I didn't do the reference site selection. That was work done by people over at Allyson, University of Queensland, and that's what I used when I did my sampling. But in hindsight, I would have chosen other reference sites as well to look at that heterogeneity issue. Yeah. Yes.
Right. I'm also feeling really ignorant about termites. Even if you were able to find grapevine and extract great eDNA from termites, do we have any reason to believe that they would be good indicators of disturbance or different type of high habitats or ecological?
Yeah, well, there has been some work exploring their usefulness, but they're just so understudied in restoration ecology that hasn't really been precisely looked at yet. I think it was more that Eris had identified them as, or potentially key bioindicators. So the fact that ants were already key indicator taxa, like termites was a nice add-on to that, but it wasn't like essential just yet. But I feel like it could potentially be a good indicator because they do so much nutrient cycling in the environment. Like ants and termites, I should have led with this, ants and termites make up a really large biomass of invertebrates or terrestrial invertebrates on Earth. Even just terrestrial biomass, it's something close to 50% if you take away any farmed animals and you just look at terrestrial biomass. So it's quite a significant amount of species, and even in a mound you can have over 1 million termites. So they're really abundant, and they do a lot of decomposition and help to cycle nutrients, which I think is really important in restoration ecology, and something that maybe could be separate. So rather than using species composition as an indicator, maybe just looking at other I guess factors that termites bring to the table.
Just occurred to me as you were showing that, you seem pretty on this. Like, yeah, would we expect them to show much distinct? But yeah, no.
Yes, I think Harris's thoughts on including were this. Yes, the successional trajectory as after disturbance, we've got no idea. But there's an assumption that if we're having a properly functioning ecosystem at the end, it needs to have the termite communities there. And I think that's what we're Whereas with the ants, we've done a lot of work on looking at successional changes of disturbance. That's not great. Righto. Well, thanks everyone, and thanks again, Ally. It's been an absolute joy having you as a student. It's a long time ago, wasn't it? It's October 20. So please join with me in thanking Ally for such a fabulous thesis, but a fabulous presentation as well. We'll know, but will it be in past? So please. So you over there.
Yeah, my job, Ally. I just wanted to give a little gift to Ally, so thank you very much for your seminar today. It was fantastic, and I think you really walked us through the development of those methods into then applying them in the environment, which is fantastic. So thanks very much. Thank you. And our next seminar will be in a fortnight, and we have several speakers who will be joining us from the NT Herbarium. And please join us for morning tea at 10:30 in the Yellow 2 tea room.